Hello and welcome back to our series. We're grateful you're here. Before we introduce today's session, I wanted to remind you that you can find our updated programming and useful resources on the web, including a new one-page bladder cancer patient navigation aid designed to help our patients understand the complex relationships between our unique teams and the departments across the GBCI. You can download a copy from our website landing page or email us at bladdercancer@jhmi.edu to request this valuable tool. We are always expanding our educational opportunities, including our growing collection of programming led by Drs. Armin Smith and Jeannie Hoffman-Sensis and program team members Pamela Getz, Samantha Rockler, and Stephanie Cooper Greenberg, specifically tailored around the experiences of women with bladder cancer. See our website for the next virtual women's group. Shortly, we will be featuring our offerings for next semester. All relevant details can be found on our website. Simply Google Greenberg Bladder Cancer Institute. Again, more information for all of our programming can be found on our site. Just Google Greenberg Bladder Cancer Institute. And if we can help you directly, we are only an email away at bladdercancer@jhmi.edu. I'm grateful to all of you for joining us today for a conversation about bladder cancer and the microbiome, understanding complex interactions. Today, we're joined by two spectacular co-hosts and strong advocates of our institute, Dr. Cynthia Sears and Stephanie Cooper Greenberg. Dr. Sears is the Bloomberg Kimmel Professor of Cancer Immunotherapy, a professor of medicine and oncology at the Johns Hopkins School of Medicine, and a professor of molecular microbiology and immunology at the Bloomberg School of Public Health. She completed her medical school training at the Sidney Kimmel Medical College of Thomas Jefferson University and completed residency and fellowship in internal medicine at New York Presbyterian Weill Cornell School of Medicine. She completed additional training in infectious disease at Memorial Sloan Kettering and at the University of Virginia School of Medicine. She's an expert in foodborne and enteric infections and has studied the pathogenesis of enterotoxigenic Bacteroides fragilis, ETBF, both in the laboratory and in clinical settings for over 20 years. Her laboratory is currently performing both mouse and human studies to define how the colonic microbiome controls colonic inflammation and colon cancer. Today, Dr. Sears will start by providing us with an overview, and then we will be having a discussion. Please feel free to type your questions into the chat.
Oops. Good afternoon, everyone. Thank you for joining. And as Dr. McConkey said in that very kind introduction, I'm Cynthia Sears. I'm an infectious diseases specialist. And today I'm going to talk to you about the microbiome in health and disease, and then also to bring it back to bladder cancer, which I know is important to all of you. I do have a few disclosures, research grants from industry. I'm on an external advisory board, but it hasn't started yet. And I do some reading and reviewing for different journals. So this is what I hope to cover this afternoon. And first, I'm going to talk to you about how we define the microbiome, why we think the microbiome is critical to our health, what modifies the microbiome, what about our microbiome in disease and in therapy, what about yogurt and probiotics, and then how I'm thinking about it looking forward. So our microbiome is integral to all of us, and it's all around us. So every surface of our body and every contiguous, what we would call mucosal surface, is covered with many, many microbes. So there are words used in this field. One is microbiome, and that actually is referring to the genes in all of the organisms we are carrying, and microbiota, which is actually the different bugs that are in each of these communities. So we share our bodies with about 100 trillion, just a huge number of microbes that can be bacteria, viruses, fungi, or parasites. And as I said, they're on every external and contiguous body surface. At this point in time, and this has really been true for the first 20 years of the microbiome field, we know the most about bacteria because they're the easiest to identify. And we think about all of these organisms in three groups. One are called commensals. So they just sit with us. They don't really interact with our host cells. There are mutualists or symbiotes, and those are ones that interact with our human cells and in a collaborative fashion. And then there are, of course, some microbes, really the minority, can hurt us, which is what we call pathogens. So this is one of my favorite stories in biology. So I'm going to tell you about a system that was described from 1970 into the 1990s, and why this is such an interesting story. So at the top, let's see, do I have an arrow? Yeah, there it is. So in 1970, Dr. Hastings was working in his laboratory and grew up an organism. And he realized when the culture got dense with the bacteria, all of a sudden it glowed, it created light. And eventually that became an entire field in microbiology called quorum sensing. And the importance of that is this is how bacteria talk to each other. They create small molecules or chemicals that actually influence each other and change the behavior of their neighbors, which is why we think communities that we carry on our skin or in our gut are really important to our function. Fast forward to the 1990s, and this paper in Science identified that this little animal called the Hawaiian bobtail squid, which we knew filtered huge amounts of seawater, and seawater is loaded with microbes. But it turned out that only one sticks to these pictures in the middle, which are called scanning electron micrographs, to this organ in this animal. It's called the light organ. And when only one bacterium sticks to this organ, and you can see how it changes shape over the 18 days that these pictures display. And at the end of those 3 weeks, now the little organism, the bobtail squid, can glow. And it turned out that the organism that sticks to the light organ was exactly the same bacterium that Dr. Hastings studied. So you would say, why would this little animal want to glow? And it turns out it was a defense mechanism. So by glowing, the organism at the bobtail squid at night floated in the seawater, and its predators would look up and would not be able to see it because it blended with the sky. So to me, this is a beautiful example about how one bug can make an incredible difference in the story of an organism. So from there, I'm going to tell you why we think the microbiome is important in health. So each of us on this call have our own personalized microbiome. So the microbiome is hugely diverse, but it turns out that this group of bacteria possess similar functions for each of us, which is a concept called functional redundancy. In other words, we each have our unique communities, but they're sort of doing the same thing. So lots of bugs can contribute to health. The second reason why these organisms are so important is because, as I mentioned, they're sometimes symbiotic. They work with our human cells to promote good function of a particular tissue. That's particularly true in the skin versus the gut. In the gut, we know that the microbiome improves our efficiency of digesting and absorbing nutrients from the food that we eat. This was actually shown in the 1930s when mice were made germ-free. And germ-free mice eat all the time, but don't gain weight. But when they're given a gut microbiome, then they eat less, but they gain more weight. So we know the bugs are very important to our nutrition. And lastly, they're very important to internal organ development, like tissue development and our immune systems. Because these principles were being uncovered about 20 years ago, the NIH realized we needed to know more because we really didn't understand these important groups of bacteria. And so the Human Microbiome Project was begun, and it focused on studying 250 individuals at 5 body sites over a period of time. And the real goal of this project was to define what we call the ecology of the human microbiome, to define what was in our mouths, what was on our skin, and what was in our gut. And it turns out our largest group of bacteria or microbes is actually in the gut, and second place is the skin. So what modifies our microbiome? You know, we talk about good microbiomes and bad microbiomes, but even to this day, we don't know exactly what a good microbiome is. There are 3 major processes that can modify the microbiome. One is what we eat. The second is the medications we take. And the third is genetics, what the genes in each of our host cells are doing. I'm not going to talk much about genetics today because that's considered a pervasive effect on our microbiome, but one that's much harder to define. In contrast, we can see big effects with diet and medications. So I already mentioned from the 1930s that mice changed what they ate based on their microbiome. And on this slide, I'm just showing you 3 papers that I think were so important to helping us understand how diet influences the microbiome. So the first paper in 2011 that I have here was a study of diet. And what they did was they took individuals and they gave them a vegetarian diet or they gave them a meat-based diet. And what they identified was that within 24 to 48 hours after changing your diet, your microbiome changes. So our microbiomes are very responsive to what we eat. And it also says that if diet can change your microbiome that quickly, we are actually empowered to improve our microbiome and probably its function based on what we eat. The second paper in the bottom there in 2015 was a remarkable study done at the University of Pittsburgh, where they took individuals, African Americans here in the United States, who mostly had a high fat and low fiber diet, and compared them to African individuals in Africa who had mostly a high fiber and low fat diet. And after figuring out what that baseline was, they actually switched the diets. So the African American diet was what was given to individuals in Africa, and the African diet was brought to the individuals, the African Americans. And indeed, in only 2 weeks, they could see a big shift in the metabolism of these individuals. Again, reinforcing that we are in control when we eat our diet on how our metabolism is working. And the third paper, also published in 2015 in Cell on the right, was a study done in Israel that was a massive study. Studied lots of individuals in exquisite detail. And the most interesting thing I thought from this paper was that each of us is different in the sense that one person can eat pizza and have no impact on, say, their blood glucose or perhaps their tendency to have diabetes. In contrast, a second person with a different microbiome can eat pizza and end up with a high blood glucose. So I can't emphasize enough how important diet is to our health. Second in line are the medications, and probably the most important medications modifying the microbiome that the vast majority of us have taken more than once probably in our lives is antibiotics. And one of the issues as an infectious diseases specialist, one of the issues we face is what we call inappropriate use of antibiotics. Antibiotics only treat bacterial infections. They do not treat viral infections. And a huge percentage of antibiotics that are used are not used well. In addition, this is a source of antimicrobial resistance issues in this country and elsewhere. So antibiotics definitely save lives, but they do have adverse outcomes, and some are listed here. I've mentioned antimicrobial resistance. Another outcome that you may have heard of is something called C. diff or C. difficile infections. There can be allergic reactions, and toxicities like nausea, vomiting, diarrhea, or vaginitis are actually pretty common with antibiotics. And as I mentioned, the antibiotics unequivocally change our microbiome. And there's a debate going on about how durable that is. It used to be said that a course of antibiotics, we would reverse those effects on the microbiome within a couple of months. And now that's much less clear. In fact, there's now data, several studies that suggest distant antibiotic use 10 years prior to the onset of colon cancer actually can modify how frequent colon cancer is. So there's concern that antibiotic exposure may in fact be linked to illnesses that are not infectious, but are what we call non-communicable diseases, things like heart disease, things like colon cancer and other cancers. So if we turn to talk about diseases, I can tell you that every body site is under study, and everyone's trying to, all the researchers in this field are trying to identify the linkages between the microbiome and disease with the hope that we can modify the microbiome and improve either the outcome of disease or the outcome of therapies. And some of the really interesting ones are listed here, include obesity and malnutrition, atherosclerosis or heart disease or hardening of the arteries, as well as cancers, as we're going to talk about in a moment. So we already know that there are a group of microbes, many of them viruses, that underpin cancers. And in fact, about 25 to 30% of cancers around the globe are thought to be related to a chronic infection by a single microbe, most often viruses. So the organism in gastric cancer is Helicobacter pylori, and is thought to be related to about 90% of gastric cancer, and about 10% of gastric cancer is thought to be related to a virus called Epstein-Barr virus. Liver cancer is related to the hepatitis B and C viruses, cervical cancer to human papillomavirus, as is anal or penile cancer. Bladder cancer, mostly in Asia, is linked to schistosoma hematobium, where schistosoma parasites are more common. And biliary cancer to two other parasites, again, mostly seen in Southeast Asia. Now, as Dr. McConkey mentioned, I've been working in the colon cancer field, and I'm simply using this slide to sort of illustrate the range of ideas that are being pursued in colon cancer and in other cancers. So on one side is the idea that there are single species, as I just mentioned, or single microbes that are important to disease. And here I've listed several bacteria, each of which has a pretty substantive connection to colon cancer in humans, including the one that Dr. McConkey mentioned that we've been working on for a long time, and that is the enterotoxigenic Bacteroides fragilis, or ETBF. And so in the picture, this is a mouse. What you can see is in the control mouse, you don't see all these dark tumors. And what this picture illustrates is we can add this single bacterium to this mouse and indeed foster the development of tumors. On the other side of the equation is what we think about as microbial communities. So going back to Dr. Hastings' organism and the glowing bobtail squid. In our case, in the gut, we think about biofilms. And biofilms are communities of bacteria, as shown in this cartoon, that are invading this gray space I'm showing there, and they're now sitting right on top of those epithelial cells that line our gut. And what this is happening here is these bacteria possess the capacity to invade what we call the inner mucus layer, which lines our entire gut and is our host defense. This is normally sterile, and the bacteria cannot get into this layer. But once they get into that layer, they actually can change the function of the cells in our gut, and in fact, precipitate cancers or tumors in the gut. It also has been a way that we've identified new specific contributors to colon cancer. So on this list are many cancers that are suspected as being promoted by the microbiome. And by that, I mean, we don't understand that they actually cause the mutations in the cells that are so critical to cancer. But we understand that if these microbes, if particular microbes are around, it can make the cancers grow and progress more quickly, or at least that's what we think can happen. And this, if you look at this list of cancers, these are all cancers that have access to the environment and microbes. The exception that I put in parentheses there is liver cancer. It sort of has a connection to the outside, but it's pretty distant. And in general, these are all, I would say, understudied areas of the cancer microbiome interface at this time. So as I wrote here, the list is longer than actually the evidence. And I did want to point out that breast cancer research in the microbiome is being done here by Dr. Dipali Sharma at Hopkins. So I wanted to comment specifically on bladder cancer. And I had looked at this a few years ago, and I took another look in preparation for speaking to you today. And this is what I would call a thin field. So there are a number of small retrospective, which means looking back studies. And what you can identify is that the microbes associated with tumors or the normal tissue in the bladder, if you're a man or you're a woman, or if you're a person who is treated for bladder cancer with BCG, so responders and non-responders, there are variations seen, but consistent, clear results are not yet apparent. However, what is likely apparent is that the microbes plus what we call inflammation, so the immune response to the microbes that creates disruption in the tissue, is probably key to the genesis of cancer. It is interesting that the bladder cancer risk factors are also risk factors for a change in the microbiome. So as we age, our microbiome does change. Smokers have a different microbiome, not only in their lungs, but even distally. Like, for example, in the right colon, smoking can change the microbiome in the right colon. And then those individuals with recurrent UTIs, as I understand it, and Dr. McConkey may correct me, but that these types of events, these infections, can create low-level persistent infection. And furthermore, more recently, it looks like about 20% of urinary tract infections are related to infections with what we call a PKS positive Escherichia coli. So this is an E. coli urinary tract infection that has particular genes. The PKS stands for polyketide synthase. So this is a group of a dozen genes that are present in the E. coli that make a particular molecule called colibactin that actually can damage DNA. And these organisms have been increasingly studied in recent years and are being linked to colon cancer, and similar processes might be true in the urinary tract. I think a really great question is whether or not the microbiome impacts responses to BCG or other immunotherapies that are being used in urogenital tract cancers. And so I think it's a really good question whether or not the urinary microbiome or the microbes that are in the urinary tract might be affecting responses to BCG therapy. But I don't think we have that answer as yet. And just to extend it, we do know that immunotherapy responses studied more in lung cancer and melanoma are thought to be influenced by the microbiome. And lastly, when there's persistent infections in the urinary tract, the question is raised about whether or not there's biofilms. There's those clusters of bacteria that I mentioned that might be changing the function of the cells. So I think what's needed in this field is really a prospective, well-designed study. So I did want to mention immunotherapy as important to so many cancers now. And it's been thought for now several years that the gut microbiome might be influencing how effective immunotherapy can be. As we know, immunotherapy has been a remarkable breakthrough in cancer therapy, but only a minority of patients in most cancers respond. So it's on the order of 15 to 30% overall, and that's just too low. So there's a lot of work going on to try to understand if we can manipulate the microbiome such that it actually can improve the outcomes to immunotherapy. So how might we change the microbiome? Well, I've already, I hope, convinced you that diet can be very important. Another way that is discussed is with supplements such as probiotics, or there's also prebiotics. There's a couple different formulations of organisms or substances that can be taken that will modify the microbiome. I suspect you've heard of fecal microbiota transplants. This is taking the stool, for example, from a patient who has responded to cancer immunotherapy and put it in another individual. These types of experiments have been done and show some response or some improvement in response. And lastly are engineered microbes. And there's an increasing number of companies that are engineering microbes to make compounds that they think can improve the function of the microbiome. So I always like to bring up probiotics. They're quite popular. These are not considered drugs. They are dietary supplements or medical foods. They are not regulated by the FDA. They actually fall under an agricultural definition. Now, probiotic technically is considered a live microorganism, which when administered in adequate amounts, which is oral, confer a health benefit on the host. Whereas prebiotics is taking a supplement that actually is supposed to promote the growth of good microbes in your gut. So probiotics are extremely variable. It's one of the biggest money makers on the planet. They can be single bacteria, multiple bacteria, or different types of bacteria. However, I think the consumer needs to be aware there is zero quality control in these products. There's no regulatory oversight. They're considered generally regarded as safe or GRAS. There are very few well-done studies in this field, and only 2% of studies report safety measures. So there is, at this point in time, no FDA-approved product for any condition. So I think it's important for people to know that these are not proven to be helpful. And in fact, some data is accruing that they can sometimes be harmful, including in immunotherapy. And this is the major paper published just a few years ago in Science, looking at the effect of probiotics and dietary fiber on the microbiome and outcomes in immunotherapy in individuals who had melanoma. And I'm just highlighting this one phrase: the best benefit, most pronounced benefit, was observed in patients who took enough dietary fiber and did not take probiotics. So this may not be the final word, but I think it is a cautionary note in terms of deciding to take probiotics when you're under cancer therapy. In contrast, I encourage yogurt and kefir. So all yogurt has 2 live, what we call lactase-producing bacteria that are shown here. And then more live bacteria are added to yogurts in the store that are labeled probiotic yogurts. Kefir is an ancient product. It's a fermented milk drink originally that's complex, and it actually includes both live bacteria and yeast. Yogurt is considered a nutrient-dense food that may help calcium absorption. And in contrast to probiotics, there is no evidence that eating yogurt or kefir is unsafe. So a few final words. This microbiome field is still a very new scientific frontier, but it is one of increasing promise. I suggest to patients that they protect their microbiome by eating a healthy diet, exercise. There's increasing evidence that exercise actually modifies the microbiome, and there are studies going on in both hereditary colon cancer and breast cancer in particular, suggesting that exercise actually improves survival under those conditions. Anytime you're asked to take antibiotics, you should ask why. And just remember that if it's a virus, the average cold, antibiotics do not help. Probiotics are not of proven benefit, but yogurt and kefir are low-cost foods that may benefit health. And times are really exciting in this research. In 2023, we now actually have 2 FDA-approved microbiota modulating therapies. One is called Rebiota, and that is actually it's like a fecal microbiota transplant, except it's developed with quality control, which is not true of the average fecal microbiota transplant. And a drug called Valst, which is an oral microbiome drug that is actually consists of very common bacteria that we all have in our guts, and it's a way of repopulating the gut with, we think, good bacteria. And with that, I will stop and be happy to answer any questions.
Thanks, Cindy. That was fantastic. And thanks for plugging the project that we started with you under the direction of Dr. Armin Smith. The postdoc Yujiro Hayashi's actually just generated a whole bunch of rectal swab microbiome libraries for us in there. So again, I agree with you. I think a lot more work needs to be done with urothelial cancer. So I'm going to turn it over to Stephanie to serve as our ringleader for the discussion period. Stephanie?
Well, first of all, thank you, Dr. Sears. That was fantastic, and I have lots of questions. But first, I have one for Dr. McConkey. Explain the study of
well, so there's a lot of frankly nonsense flying around right now, but what might be going on with regard to the interaction between the microbiome and bladder cancer. And so together with our colleagues at other institutions and also with Cindy's group here, we're exploring exactly what the microbiome looks like, both in the GI tract and in the bladders through the urine and urine sediment, patients without and with bladder cancers. And we're performing what we call longitudinal studies, meaning we're making collections as Armin treats these patients with BCG. And we're thinking about gender, and we're thinking about diet. We've thought quite a bit about how we would modify the diet. And last year at the Bloomberg Kimmel Cancer Immunotherapy retreat, we heard some pros and cons about how maybe difficult that is to do, because it's hard to control what people eat unless they're inpatient. But we've thought about supplementation of some sort, either with yogurt or something else. And so I think step one is to see what happens at baseline. And then step two is to see if there's something that consistently happens when we try to intervene with a supplement of some kind. We have a friend, Monty Paul, who actually works at City of Hope and does mostly renal cell carcinoma research. But he published a fairly high impact paper recently showing that another oral supplement increased response rates to immunotherapy in patients with renal cell carcinoma, published in Nature Medicine. He's actually talking to us about that study. We've been in touch with the company who are making this supplement for distribution here in the US. It's currently not available in large quantities. But again, cautiously, we're thinking about this. There's a good example in bladder cancer of how taking remedies, homemade remedies, can cause problems. And that is, in China, there are a lot of herbal remedies that patients have taken that have actually caused their bladder cancers. We can tell that by the genetic signatures that are present in their tumors. The substance is called aristolochic acid. It's not a bacterium or a virus. But again, it's not necessarily true that you can't hurt yourself by taking extra things that you think might be beneficial to you. So we are gradually and cautiously ramping up our own efforts to study the microbiome in patients with bladder cancer.
Got it. Hippocrates said that is known to have said all disease starts in the gut. It seems to be the case here today. And I have a question. We're curious, and it's leaning toward, with more study, that there may be, as your work with colon cancer, more causal relationship between the microbiome and bladder cancer, especially because it's an epithelial cancer. But in general, could you say that the microbiome, a healthy microbiome, whatever that means for that one person, can also prevent disease? Does it work against? Is it a good warrior within you to sort of prevent cancers from happening?
Well, we certainly think that's true. It's unclear, I would say, even in colon cancer, which has been studied the most so far, whether the organisms are causal or are responsive to those original mutations that occurred. One piece of information that I've been puzzling over is that in the gut and even in the lung, they're now identifying that some of these gene mutations that are critical in some individuals to development of cancer are happening in normal epithelial cells. So they're not cancerous cells, but they're normal cells that have these mutations. And it's very unclear what the history over time of that is. Do they just fade away? Do they start to change the biology and so much later turn into a cancer? I think, and is something restraining them, and what's restraining that situation? Most of us are developing mutations all the time, and we're clearing them. The body is pretty robust in that regard. But when conditions are right, and I do think this is where the microbiome could contribute, and the balance is off, then maybe the cells can grow more robustly and create a tumor.
I think we're going to have another one of these sessions focused on what Cindy just said, which is that in me and anybody else over the age of about 50, we know there are age-related mutations that occur in the bladder. And so, as she said earlier in her talk, that she thought that the microbiome was acting in some cases more as a cancer promoter. We think of initiation as the thing that happens that causes the mutations. And promoters are the wide variety of other things that help promote cancer growth. And so another thing that we're doing, as you know, is we've set up a kind of a toxicology program under the direction of Sunil Patel to see if some of the environmental contaminants that he's interested in actually promote these small patches of normal cells that contain mutations, promote them to switch in some cases to become cancers. So I think it's a subtle distinction and one that we'll have to get into at a future date. But I agree. I think in both cases, lung, colon, and also bladder, we've got these initiated cells that might be strongly influenced by the microbiome.
Fascinating. I have a question I've always wanted to ask. So certain types of subtypes of bladder cancer, particularly squamous cell, is as your slide indicated from I hope I say this right, a schistomal infection. So everybody's unlucky when they get cancer and super unlucky when they get bladder cancer. And we want to dispel those myths that, it was your fault. But I've always wanted to ask this question, my husband had squamous cell bladder cancer. He's a businessman. He's never been in a jungle. He doesn't drink from water fountains. He doesn't do anything extraordinary or unusual, but he had squamous cell. Where would someone acquire a schistomal infection? Is that hereditary? Is it from water? Where do you get these things?
It's from water that's
Oh.
infected, right? So in the United States, the schistosoma hematobium is not native to the United States, but there are schistosoma. If you go up to the Great Lakes and you decide to splash around in the water, there are people who will get an itchy rash on their feet and ankles. And that's because there's these schistosoma that penetrate. It's a very complicated life cycle, but these little parasites that can penetrate the skin and then irritate. And that's what causes the rash. But the ones that we have in the United States can't go any further. The ones in Southeast Asia where hematobium, there's several of them, Hematobium mansoni and so on, japonicum, there's several of them, have a different capacity to penetrate and disseminate. And therefore, depending on the type of schistosoma, they like to either go to the bladder veins or they go to veins that are around the colon or rectum. And so you get different types of disease depending on the type of schistosoma. But it's a regional acquisition. So I wouldn't, just like the parasites, I can say them, but I stumble a little bit with the ones that cause biliary cancer, also an epithelial cancer, but almost always had someone who'd been in Southeast Asia or grew up there. And then these become very chronic. And it's that chronic carriage and persistent inflammation that the parasites cause that actually are the trigger for converting to a cancer.
Wow. Okay. I have tons more questions, but we're going to go to our wonderful audience here. So Marilyn is asking, her question is, can bladder cancer be caused by a previous GI infection?
That's not generally, I would say, although we know there's a linkage between what's in the organisms that are in stool and the occurrence of recurrent urinary tract infections, particularly in women. And so it turns out the PKS E. coli, which are getting a lot of attention now, do not cause diarrhea. They are not your average cause of foodborne disease, which all of us have experienced at one time or another. They're actually silent actors. They're there, and they can be doing damage depending on the circumstances, I think, in the gut. And they can get into the urinary tract. And there are also other, what we call gram-negative rods, like Klebsiella, that can actually pick up these gene sequences that make the same DNA damaging toxin. So we know that more than one common organism that you see in recurrent urinary tract infections actually could be what we call potential inducers of DNA damage, and hence potentially cancer.
Wow. Okay. Karen asks, can you talk about gender differences in the microbiome?
Not well, I have to say.
That's a nice setup too, because that's one of Armin's interests. So
Yeah.
Yeah.
Armin and Jeannie Hoffman senses. So yeah.
Somewhat, this is sort of the difference. There are definitely hormonal influences, and I honestly don't have a catalog in my brain because it varies in the situation. So I will turn that question over to David.
Yeah, so we don't really know, Stephanie and colleagues. I think that there's maybe, as Cindy convinced you during her talk, there's a lot of complexity. It's not like we all have the same communities. And so it's really hard to tell if there's a gender difference given all the diversity. So we're trying our best, again, also with colleagues at other institutions, to measure this in larger numbers of people. And then together with experts like Cindy, evaluate whether we're seeing patterns or not. But we're interested in the possibility, because certainly there's a big gender difference in incidence and aggressiveness of bladder cancer. So anything that might be gender related, we're interested in.
It's Dr. Sears, it strikes me that your field is just full of questions, and it seems like it's been understudied and maybe undervalued as a causal or a companion factor in all sorts of things, just basically people not feeling well or having, infections or having to take antibiotics, and then it goes up and down the food chain, no pun intended, there's colon cancer and all. Do you see a time when there's more, you're on the front lines of robust research, is this field of microbiome research as it relates to cancer specifically growing, and all these answers maybe in time could be revealed?
Yeah, I've become of the opinion that what's really important is to do human studies. There are a lot of biotech companies, including one we're talking to now, that are making really what we call probiotics that are engineered. So it's a particular bug that we know is safe for people, and then engineered to produce an enzyme or something else that we think positively influences the microbiome and might improve the response to immunotherapy. I really personally feel we can, I think mouse experiments are important. I don't think they're directly telling us what we need to know in humans. We really need to do the very hard work and the very careful work of making sure the probiotics, if you want to call them that are being studied are safe, and then to study carefully how they're influencing the biology and outcomes of disease. But it's a slow process. It takes a lot of time to do this.
I'm sure when you started out in infectious diseases, you never sort of imagined that you'd be where you are today, with the leading expert with microbiome and immunotherapy, which didn't exist, I assume, when you were in Thomas Jefferson. Is that right?
You're totally correct. I thought I was going to be a global health diarrheal disease expert.
And we're so glad you didn't stay there. A few more questions. Two from similar questions from Vietnam vets. Thank you for your service. the role of Agent Orange and bladder cancer, or Agent Orange and its assault on the microbiome. Any thoughts on that?
It would not surprise me at all if there was an impact. I haven't seen studies on that. There may be some, but I don't know the exact answer to that question. It's a very good question. Thank you for the
We're
question.
interested in answering that question too. Dioxin itself is really stable. I worked on it as a graduate student, and it's dangerous to work with. And so we're going to study compounds that activate the same pathway in part of Sunil's project. So whoever asked that question, stay tuned.
Right. Cindy asks, I'm a bladder cancer patient who underwent 15 BCG treatments, and we all know BCG is a live bacteria. I've read that turmeric or curcumin can be beneficial in improving response to BCG during treatment. Would you agree?
I am aware, I haven't seen those studies in bladder cancer, but maybe David has. I know in the colon, the same theory existed, but it was proven to be negative. The studies did not show benefit when curcumin was studied.
Interesting.
Yeah, I'd agree. I haven't seen any evidence for that. When I used to work on curcumin many years ago, we did a lot of research on this. In fact, one of my colleagues, Rizal Kurzrock, did a clinical trial, and we really didn't see a signal there. So I think that there may be better ways to help your microbiome than taking curcumin.
Okay. Takeaways about diet: healthy diet, Mediterranean diet, exercise. Those are pretty much the
Those
standard.
are, those fundamentals, which actually are promoted by the US government. In fact, you can go online and find all that information, including long lists of foods and how much fiber is in them. These are very important concepts. For example, fiber, when worked on by bacteria, releases these compounds called short chain fatty acids, and short chain fatty acids are the #1 energy source for the epithelial cells in the gut. And so while most of the literature is a little complex on everything, in general, short chain fatty acids are thought to be good for our microbiome and for the health of our gut.
Okay. I have a question, and we'll get back, then we'll tee up Dr. McConkey and his work and the GBCI's work on biomarkers and its connection to the microbiome. But specifically for our beloved bladder cancer patients who have to undergo a urinary diversion, that's a pretty big assault to the gut and to the balance within the gut. And I don't exactly know what I'm asking, so forgive me, but is there a sense that this enormous surgery or this rerouting within the gut causes damage, and does the gut reset? Are the good bacteria working hard to make things go back to normal after a surgery, say for colon cancer or bladder or whatever abdominal surgery you may have? What is the role of the microbiome with things that go wrong or the unfortunate surgeries?
Yes, and the other day when we were, organizing to be here today, you asked that question and I realized I didn't know. So I tried to look it up, and I actually found no studies that have really addressed that. I completely agree with you that is a huge physiologic change. And so you would expect some impact, but I don't think it's been studied unless David knows something.
Well, the good news is we're giving David McConkey a long wish list of research projects as a result of this, because we're realizing there is a lot that can be studied. A visiting fellow from MD Anderson, Dr. Steinmetz, is with us, and she's going to ask the question I was going to ask about the correlation between the urinary microbiome, that would be urine samples. The dream is to have a urine, we all have urine, sorry, it's a topic we all talk about around here. Wouldn't it be great if the urine could tell us what's going on with those who've had bladder cancer? Is it predictive? Is more coming on? Or is everything stable, etc.? What is the connection between the urinary microbiome and the gut microbiome? And David, please pipe in as to your work about.
Well, she's going to have to help us study that now that she's brought the
Okay.
question up. So,
Done. Top of
yeah,
the list.
let's let Cindy go first.
Well, it was hard for me to discern that it was clear, first of all. And there is this low-level urinary microbiome that's discussed. And I've always been interested in Scott Holgren's work at WashU, which has shown that there's some organisms that sort of go silent, but are there on the surface of the bladder and probably changing function. And we know that that's something that can happen with other bacteria. For example, cholera in Bangladesh during certain seasons of the year can be seen in the water and identified in the water, but it won't grow. And then conditions change and it can bloom, and that's when you see cholera outbreaks. Now, I'm making a broad analogy there, but I do think that understanding the urinary microbiome and any changes, and I'd love to hear what David has to say about biomarkers, because I think this is precisely what we need, is some way to identify people who are in trouble. It's true and definitely in colon cancer, and it's probably true with other cancers, an early warning signal.
Yeah, so Alexis, we do know that the immune cells in the urine in patients with bladder cancer are more similar to the immune cells in the tumor than they are to the immune cells in the peripheral blood. And so we're hopeful that we can use the urine to measure the immune microbiome interactions that may be present there. We've got, as you can tell, a lot of interest in how the microbiome regulates the immune system. And so therefore, there's obviously a chance that the gut microbiome is having as much or maybe more influence on what's going on in the immune landscape in the bladder. And so again, it's all about making accurate measurements and doing things in people that we think will change the microbiome in either hopefully a positive way, because there's a lot of correlation right now, but we really don't know if the urine microbiome is a readout of what's going on in the tumor. That's what we're hoping Cindy can help us study.
There's so much awe and wonder about even this conversation today, because when you think about, all the things that you've discussed, the millions and millions of bacteria that live inside us and do things or prevent things, or hopefully don't cause bad things. It really is remarkable that it's an incredible system, isn't it? And I imagine with the work that you do, Dr. Sears, that you're just sort of, it's a daily dose of awe and wonder because stuff happens and it's been understudied, but I could see a day where it will be more sort of known and maybe helpful in the war against cancer, certainly, and hopefully. And that what is your prognosis for my last question for the microbiome? Is there any way that it could be beneficial in the immunotherapy field, since immunotherapy is here to stay and it's seen to be positive? Do you see a day where both can work together and really be sort of build a super immune system?
I certainly hope so. That is part of the goal of the Bloomberg Kimmel Institute as well, is a way to, within that institute at Hopkins, I'm heading up the microbiome program. And this year we're really pivoting after some studies done both in humans and mice, to try to begin to test if we can modify the microbiome and show positive signals that will promote better outcomes for patients with immunotherapy.
Wow, that would.
So as everybody knows, I guess, the rules that govern response to immunotherapy seem to be very similar in different diseases. So melanoma and lung cancer, the rules seem pretty similar to what we see in bladder. So we hope that we can leverage the information that Cindy and her colleagues are gathering and individuals elsewhere, like Jen Wargo's group, to really understand how we can maybe modulate or exploit the microbiome for better outcomes in patients with immunotherapy.
I want to just ask this last question because it's important to the person sending it. And it's a giant question and we have like 30 seconds left, but why would Agent Orange be presumptive for bladder cancer, but perhaps not for colon cancer? And is that true?
So Agent Orange binds to a receptor called the aryl hydrocarbon receptor. And we have evidence from bladder cancers that the aryl hydrocarbon receptor is important for bladder cancer growth. So there is a bit of a smoking gun, and in fact, there's a company that's developed an inhibitor of this receptor and has demonstrated some clinical activity in patients. So I think that there seems to be a pretty strong circumstantial evidence connection between dioxins and anything else like them that activate this aryl hydrocarbon receptor and bladder cancer promotion. Again, we don't think it's damaging the DNA, but it's probably helping the cells that have mutations grow better. So this is a really, really important question. Again, it's been difficult to study Agent Orange / dioxin itself because it's so stable in the environment. It's one of the problems with these Superfund sites. So we're going to use some substitutes that activate the receptor to really try to understand what's going on.
And one last question, just to clarify, if you have a neobladder, does that, so are you saying that checking a neobladder's patient's urine does not give you any ideas how the person is doing? Maybe not yet, Sam, but that's what they're working on. Is that correct, David?
Yeah, we don't know exactly what the relationship is between some of the diversions that our urologists are using and the microbiome. And, these are questions that we need to do experiments in people to answer. Again,
List, please.
doing the right measurements with the right
Right.
people overseeing
Fantastic
us.
question. And
Yeah.
we are at the top of the hour, and Dr. Sears, we could go on for 3 or 4 hours here. And I know you're giving another talk later tonight. This has been full of awe and wonder, as I mentioned before, and a really thoughtful, really exciting conversation because, David, get your pad out and start numbering these new projects. What's clear is that the day to study this is here. And let's see what we can do to start having meaningful conversations about the role of this, miraculous microbiome and how it can be helpful. So thank you for a wonderful, wonderful talk today. And thank you, David. And everyone, most of all, have happy and healthy holidays. Eat healthy food, exercise. Ugh, everybody says that. It's so boring. But we want you back next year, and we just want to make progress in this field. It matters to us. Your questions are important, and we're on a mission. We just wish we had better answers, but with Dr. Sears, we had some really great insight today. Thank you all.
Thank you. It's great.
Bye, everyone.
Bye.